Welcome back, everyone. In this video, we're going to take an in-depth look at viruses. You might be thinking, where do viruses fit into the whole taxonomic grouping of life? And the truth is they actually don't, because viruses are non-living things. For the sake of the DAT and for the sake of most biology courses, they are non-living. So that means they don't fall under the Linnaean taxonomic divisions of kingdom, phylum, class, and so on. But we should know a little bit about viruses and their key characteristics. So what we should know is that viruses are non-living things, like I said. They're parasites that infect a host cell, and they're going to use the cell's machinery to manufacture and duplicate more of itself. Whenever you see the suffix phage, that's dealing with a virus. So a bacteriophage is a virus that infects bacteria. Viruses are very simple. They're super small, that's why they can infect something as small as bacteria. They're going to be made up of either double or single-stranded RNA or DNA. It's never going to be both. It's always going to be either RNA or DNA. Now, around this genetic material, the RNA or DNA, they're going to have a protein cover called a capsid that's going to enclose the genetic information. Now, this capsid is made up of polymers known as capsomeres. And sometimes superficial to the capsid, you're going to have a phospholipid envelope, and that's obtained through the host cell membrane. So when it leaves the host cell membrane, it's going to take some of that phospholipid bilayer and enclose or envelop itself in that membrane. Here we just have some simplified anatomical structures of a virus. So remember, always deep within the virus itself, you're going to have either DNA or RNA. It's never going to be both. And that genetic information is always going to be enveloped in a capsid made of capsomeres. So here we have the individual capsomeres. And part of that protein coat, that capsid, you might have glycoproteins. These are used in surface cell recognition, so recognizing what type of cell to bind to. Right here we have the HIV retrovirus. This has RNA instead of DNA. It also has a viral envelope. This is that phospholipid envelope that it received from a host cell. Here's just a simple picture of a bacteriophage as well, where you have DNA, or you could also have RNA. There's many different types of bacteriophages. They're always going to have that genome enclosed or encapsulated in proteins. And bacteriophages, again, are viruses that infect bacteria. Let's look at the two main life cycles of a virus, and that is lytic and lysogenic. The lytic cycle refers to when a virus injects its genetic information, either RNA or DNA, into a host cell. It's going to use this cell as a small factory and hijack all the workers, all the machinery like enzymes to produce more of itself, more of the virus. And this new virus will erupt from the host cell membrane. So the lytic is dealing with eruption. So it's going to erupt from the host cell membrane and kill the cell in the process. So it implodes essentially. And because it erupts through a cell membrane, namely a phospholipid bilayer, it's going to obtain that phospholipid envelope superficial to the capsid. Now, retroviruses are unique because these are RNA viruses. So like HIV is a retrovirus. Retroviruses contain an enzyme called reverse transcriptase. So transcription is from DNA to RNA. Reverse transcriptase is going from RNA to DNA. So it's the enzyme that catalyzes that reaction. Once the RNA is converted into DNA, it can be incorporated into the host genome and go through the lytic cycle. If it doesn't go through the lytic cycle, it's going to go through the lysogenic cycle. So let's look at that. The lysogenic cycle is when a virus hides out in a host cell's genome. So that's the difference between lysogenic and lytic. The lytic is where it erupts. The lysogenic is where it's going to hide in that host genome. So the viral DNA, remember, if it was a retrovirus that had reverse transcriptase to turn its RNA into DNA, that DNA is going to be incorporated into the DNA of the host cell. When it's dormant in this condition, it's known as a provirus, or if we're talking about a specific bacterium that it's in, it's known as a prophage. So proviruses can be dormant for a long period of time, hiding out in the cell's genome. Once it receives a certain signal from the external environment, it will undergo the lytic cycle and go through that whole process where it replicates itself and then eventually kills the own cell that it hijacked. An easy way to remember the difference between lysogenic and lytic is lysogenic has the root gen in it, which is also in genome. So it's hiding out in the host genome, waiting for that signal to replicate itself and erupt from the cell membrane. Here's a picture of both the lytic and lysogenic cycle in action. So here we have a bacteriophage that has attached itself onto the surface of a bacterium cell, and it's injecting its genetic information into the cell. That's going to be incorporated within the host cell's genome. Now let's say that this bacterium is going to undergo binary fission, where it duplicates itself. It makes 2 daughter cells. So it's going to duplicate its DNA. Remember, in that DNA, you're going to have the virus, so the information needed to code for more of that virus. So that's going to get passed on to both daughter cells here in the lysogenic cycle. So that means this virus can be dormant for a long period of time throughout many different generations. Once the environmental conditions are right, the host cell will begin to transcribe and then translate the proteins and the genetic information needed for the virus. So it's hijacking the various cellular mechanisms and enzymes to produce a massive amount of the virus. Finally, the host cell will implode, releasing that virus. That virus will get that phospholipid envelope, and we begin again with a new virus that's going to infect another cell.